Hello, I'm Barbara. I'm a Royal Society Newton International Fellow here in the Bernardes Group.
Hi, I'm Hannah. I'm a PhD student in the Bernardes Group.
We are working on the engineering of reactions to modify proteins.
And I'm working to degrade a disease-causing protein in brain cancer. Our lab has 2 synthesis labs. We are in one of them. And this is where we synthesize and prepare compounds, fluorophores, drugs in order to treat cells or conjugate to proteins. And we will next go to where we analyze the compounds we've made, and finally to where we apply them in cells or biological systems. We are now in our analysis lab where our group has our own LCMS machine and HPLC machine where we purify compounds and also analyze our protein conjugation reactions. And we can also do time course studies. And I will next show you another analysis lab where we have a plate reader and can perform gels and other instrumentation.
After preparing those compounds in a synthetic lab and analyzing it in the biolab, we normally bring them in here to perform protein modification, for example, or even DNA and RNA conjugations. And we have all the facilities in this lab to analyze them and characterize them. For example, we can use the STS page analysis for Western blot, and even we have the plate reader to calculate the concentration and also to perform kinetic analysis.
Our group has recently gained access to the Unifinity lab and this whole Chemistry of Health building, which is brand new.
And I'm now going to show you some of the new equipment that we got access to.
Our group has recently gained access to this brand new facility called the Chemistry of Health building, where there are many brand new instruments such as the nanodrop, which can be used for protein, DNA and RNA concentration and UV-vis applications too. And this facility also has more high-tech plate readers that allow long time course kinetics experiments and also gel imaging instruments. We will now take you to our tissue culture facility where we can treat cancer cells with the compounds that we synthesized in the synthetic labs. Now we are in our tissue culture facility, which is where we treat cancer cells with the compounds that we've made and analyzed in our previous labs, shown by Barbara treating her cells. And I will show you where we keep our cells and incubate them. In here. And then if we've added a fluorophore, we can do live cell imaging at the microscopes.
And when we treat our cells with the fluorescent compounds, we can image them here in this high-tech microscope. Thank you so much for visiting our labs in the Bernardes Group.
If you would like to find out more, you can find us on our group website or Twitter, and we'd love to be able to meet you in person sometime soon.